Welcome everyone to my presentation, which is about our activity on laser processing of materials to create micro and nanostructures. We will see some applications in photonics, optoelectronics, and smart surfaces. And part of our work was actually performed at the VU-LRC facility, Lasers for EU facility at Vilnius University in Lithuania. So we will see that near the end of the presentation. So to start, we all know that the micro and nanostructuring of surfaces affects the physical properties of matter. Very nice examples from nature are, for example, the eyes of nocturnal insects, which have an extensive nanomorphology, which provides them with night vision and anti-glaring capabilities. But also, many of us are familiar with the beautiful colors of the butterfly wings, which are not dyes. They're not like colors from a dye. They are actually structural colors. So they have to do with the morphology of the wing of the butterfly. So in the quest of mimicking some of these properties, there are several technologies, and of course the gold standard is lithographic methods. You can do pretty much everything you want with lithography with great precision. However, this comes at a cost. You need large infrastructures, costly infrastructures, and multi-step processing with masks, alignments, therefore a little bit size limited as well. If one can relax a little bit the requirements on precision and the repeatability, maybe and uniformity, laser processing comes as a nice alternative approach. It is capable of creating quasi-uniform structures with a tabletop apparatus, not something too large or too costly. And most importantly, it is a single-step approach without the requirement of any masks or anything. So also laser processing in silicon, which we have worked a lot with in the past, gives us the opportunity to create structures either at the microscale or at the nanoscale, depending on the duration of the laser source we employ. So with nanosecond laser irradiation, we create microstructures on silicon, and with femtosecond laser irradiation, in a liquid environment, we are able to create nanostructures on silicon. What's most important about laser processing is that it's a scalable process. As we said, there is no masks or anything. So if we can translate the sample with respect to the laser beam, we can process an area as big as we want to, which is not often the case with lithographic methods, which are limited, which are area limited. Sorry, I think I pressed on something. So to move on. I will now discuss some applications, starting from some of our plasmonics applications, specifically combined with optical trapping. Optical trapping technique, the optical trapping method, maybe many of you are familiar with because it was awarded the Physics Nobel Prize in 2018 to Arthur Ashkin. Optical tweezers or optical trapping, in short, they can immobilize small particles at the focus of a laser beam, which is usually tightly focused by a microscope objective lens. So this, how does it work? Why do the objects remain trapped at the focus of the laser beam? Because let's see, if a trapped object tries to move upwards from the beam focus, then it receives forces both from the rays coming from the left, for example, which refract through the object, and their momentum changes, which means that this exerts a force of equal and opposite momentum change on the trapped object, F1. And then there are also the rays that refract from the right-hand side, and similarly, they exert a force F2 on the trapped object, and the total force from all these ray forces is a restoring force F that pushes the particle again towards the center of the trap. Similarly, the same thing happens if the particle moves downwards. Again, the total force pushes it near the focus of the laser beam. And if it moves to the side, then what happens is that because the beam is Gaussian, now the rays coming from the left-hand side are weaker intensity compared to the rays coming from the right-hand side, which are closer to the focus. Therefore, the total force not only pushes the particle downwards, but also a little bit to the side as well, and always towards the center of the optical trap. So there's always a restoring optical force. Now, all this was working very well for many years until for microparticles, okay, for particles of the sizes of microns. But then, with the advent of nanotechnology and the advent of nanoparticles, there were some instabilities in the trapping process. Mainly because the trapping force depends on the cube of the radius of the trapped object. So the smaller the object, the weaker the trapping force. Therefore, people started observing that trapped nanoparticles would undergo Brownian motion within the focus of the laser beam, and they wouldn't stay, trapped really put in place. And this gave birth to the whole field of plasmonic optical trapping, where now the idea was that instead of using just a passive glass substrate to perform the whole trapping process on top of it, now one would use an active plasmonic substrate that would enhance the optical field in the trap and stabilize the nanoparticles. And in our approach, we created these plasmonic substrates by laser nanostructuring silicon surfaces. So this is an SEM image of a silicon surface which has been laser nanostructured, and coating these surfaces with a simple gold film, copper gold film, and this simultaneously would break into gold nanoparticles because of the nano roughness of the silicon surface. So there immediately we had the plasmonic surface at hand. And then by focusing a laser beam and trapping an object above the surface, we had the plasmonic optical trapping implementation. So we tried this with the 400 nanometer polystyrene beads in solution. We trapped them on top of our plasmonic silicon substrate. And in order to measure the trapping force, usually we place the substrate on a translation stage. And then by moving the translation stage with increasing frequency back and forth, then it oscillates until the beam escapes the trap. And at the point where the bead escapes the trap, if we know the velocity v of the stage at the moment of escape, we can calculate the trapping force that was holding the bead in place. So to implement this, we have our trapping setup. We have an infrared trapping laser beam that is focused by the objective lens and creates the optical trap. And in order to visualize the particles, we employ fluorescence microscopy. So we use a UV laser to illuminate the trapped objects, and we select the fluorescence from the trapped bits, and then we can observe them on camera. And the whole thing, okay, looks a little bit like this. So here we see three bits. One of them is trapped. And we see that, let's play this from the beginning. If I could decrease a little bit the sound, maybe. So let's start over. So we see here the beads. One of them is trapped, and the other two have to move so fast. So we have to move the translation stage so fast in order to destabilize it and to push the trapped object away from the trap. And then when we stop the motion of the substrate, the bead goes back to the trapping position. So we did these measurements. And we were able to measure the trapping force, and we see here the black data points are actually what we measured above this plasmonic substrate. So this is kind of like the trapping force as a function of the distance from the plasmonic substrate. And we see that as we move closer to the plasmonic substrate, the trapping force increases exponentially. And this is only the case when we use the plasmonic substrate, while all the other data points here, or the other colors, they correspond to different variations of non-plasmonic substrates. For example, flat silicon coated with a metal coating, or nanostructured silicon but without a metal coating, or simple glass, for example. And all of them demonstrate some trapping force which does not vary when we move closer to the substrate. And in order to verify that this was truly a plasmonic effect, we also needed to observe some change with the wavelength. because it's a resonant effect, the plasmonic effect. So we changed our trapping beam from a single wavelength infrared beam to a laser system with an optical parametric oscillator, which was able to produce an output from 750 to 1,300 nanometers. And we performed the same experiments by selecting different wavelengths from our source, and we see here the results. So what we have here is the trapping force as a function of the wavelength here, the several wavelengths that we used for several distances above the plasmonic substrate. And we see that for all distances above the plasmonic substrate, always we have a resonant effect with respect to the wavelength. So there is truly a resonant wavelength for which the trapping force is maximized. And this really proves that we are dealing with a truly plasmonic effect here. Extending a little bit our studies on optical trapping, recently we have started trapping red blood cells to measure their mechanical properties. Red blood cells are very important. They circulate all around the body and they carry oxygen all around the body. They are usually very much deformable because they have to squeeze through all sorts of tiny blood vessels and capillaries in the circulatory system. And actually, several blood disorders and diseases, they are associated with red blood cells not having the perfect mechanical properties. So their mechanical properties are very important because they help them circulate properly and function properly. The way we combine now, the way we use tweezers for red blood cells is that we have red blood cells flowing now in a solution, a plasma solution, and we can trap one of them with an infrared laser beam to study their mechanical properties. And for example, here you can see on an optical microscope a red blood cell. And at some point, we are going to apply the laser beam here where the cross is. And once the laser beam arrives, the red blood cell folds. This is the first thing that the red blood cell does. When the beam arrives, we cannot see the beam because it's infrared, but we can see the effect. So first thing, they fold when the beam arrives. And then once they're folded, we place the whole substrate on a translation stage and we start dragging the substrate with a control velocity V, which means that we essentially are dragging the surrounding medium, the plasma liquid in which the red blood cell is trapped. And this means that stretches the cell as well because it is dragged against its surrounding environment. So here in this movie, we're going to see the red blood cell. It's already folded and it has an initial length of L0. And then we start dragging the substrate and that creates a dragging force on the cell, which elongates, and then we stop dragging the substrate and the cell returns to its original position. Oh, and here it is with the cross indicating the location of the laser beam that traps the cell in place. So by this elongation, starting from the initial length, then we go to an elongated position by applying the velocity, and then, we return to the initial length by shutting down the velocity. From this elongation, we can measure the cell elasticity, and from the recovery time of the cell back to its initial shape, we can measure the membrane viscosity. So we did that to study the effect of oxidative stress on red blood cells. Oxidative stress, we induced it by using 3 oxidative agents that we see here, TPHB, diamide, and PHZ. This is important because these agents, they simulate cellular aging to understand how cells age or damage by diseases that affects the oxidative stress of the cells. And we see already from simple microscope pictures, images that compare to control samples, so no oxidative agents here. The application of these agents already increases some of the damaged cells that we see here with these arrows. So these are not very healthy cells now. So we use our method of optical tweezers by elongating the cell and measuring as a function of the velocity we employ how much the cell is elongated. We use this method to measure the elasticity of the cells when we have no oxidative agent at the control measurements, and then when we have our 3 oxidative agents. So for the two of them, for the TPHP agent, we see that this treatment does not induce any elasticity changes, pretty much it's the same. The second agent, diamide, it does increase the elasticity factor, which means that it creates membrane stiffening of the cell, most probably due to cross-linking of some of the cytoskeletal proteins. So here we have a clear mechanical effect. And the third agent, PHZ, here we were not even able to take measurements of the elasticity because the oxidative stress was so profound that there was structural damage on the cell. And we see here the healthy cell and the cell after PHZ here on the right, and it was not possible to take any measurements here. So these now results on agent-specific effects on the cell mechanics, we correlate them with the oxygen species levels that we measure independently on the cells, and we find the conclusion that mild oxidative stress preserves the cell deformability, and above a certain level of ROS of oxygen species levels, then we have a correlation with cell stiffening or damage. And this is an important finding for hematology studies. So now I'm going to switch gears a little bit, and I'm going to go to the second direction of our research, which is on the optoelectronics side. And again, it has to do with the microstructuring surfaces, because in this case, we use microstructuring of silicon in order to create silicon zinc oxide microstructured photodetectors. The goal here is to use the nice properties of zinc oxide, which is a wide bandgap semiconductor, which is very good if you want to make photodetectors and LEDs and optoelectronics at the blue or UV part of the spectrum, because you need wide bandgap semiconductors. For example, gallium nitride LEDs won the Physics Nobel Prize in 2014. But the problem with zinc oxide, which is an alternative material to gallium nitride, is that it comes naturally as an N-type semiconductor, and it is difficult to make P-type zinc oxide in order to create, p-n junctions for optoelectronics. And the solution usually found in the literature is that people make zinc oxide heterojunctions by combining zinc oxide with P-type silicon, for example. So this is what we also did. We combined zinc oxide with P-type silicon, but in this case, we also microstructured the surface of silicon with laser processing in order to increase the heterojunction area between the two materials and the light absorption on this heterojunction. Indeed, we see here some SEM and EDX images of our zinc oxide deposited by atomic layer deposition on the microstructured silicon. And the EDX measurements show that zinc, which is the red dots here, indeed was conformally deposited on silicon. And then we illuminate with white light our microstructure device here on the right-hand side. And we see that indeed we have a nice response to the presence of white light, much bigger compared to the left-hand side where we have a flat device, a reference device. So it is indeed very responsive. And more importantly, the microstructure device here on the right, again, it was responsive even at infrared irradiation, which was below both the bandgaps of silicon and zinc oxide, which is not the case when we use a flat device, a non-structured device. And the reason for that is that when we laser process silicon to create the microstructures, we do that in a sulfur hexafluoride environment, in an SF6 environment, which creates sulfur states are introduced in silicon bandgap, and this creates absorption even in the infrared. So indeed, by taking now more analytical measurements across many wavelengths, UV, visible, and near IR, here on the right-hand side, we have the comparison between the response of the reference device, the flat device, and the microstructure device. And we see that for all these wavelengths, we always have a higher responsivity when we microstructure the junction. So indeed, this increased surface area plays an important role in the responsivity of the device. But what was more interesting now in these studies was a serendipitous discovery that we were not exactly aiming for, but it was very, very interesting. Was actually the isotype system when we used N-type silicon to combine with zinc oxide. And this now isotype photodetector proved to be wavelength tunable. What does this mean? It means that depending on the bias voltage that we apply on this device, the spectrum that it detects changes. When there is no bias voltage between the two materials, or when there is positive voltage between the two materials, this detector behaves as an AR/IR detector. It detects mostly infrared wavelengths and it's not very responsive in UV or visible wavelengths. But if we apply a negative bias voltage between the two materials, then it becomes a broadband detector that responds across the UV visible near IR. And if we take a closer look at this current here, where it doesn't seem to be much current, actually what we see is that we have a polarity switch. What this means, it means that at these wavelengths where current appears almost zero, it's actually not zero, it is negative. It is a negative photocurrent, and then it becomes positive for the infrared wavelengths. So depending on the wavelength, this means that for low wavelengths, the electrons move from zinc oxide to silicon in the junction, while for high wavelengths, the electrons switch direction and they move from silicon to zinc oxide in the junction. So we reverse the direction of motion of the electrons within the junction. And in order to understand how this works, we need to consider that in this junction, actually zinc oxide has 3 orders of magnitude higher electron concentration than silicon. So when we bring the two materials together, there is diffusion of electrons from zinc oxide to silicon, and this creates an internal electric field, which at some point opposes further diffusion and stabilizes the junction and the built-in field in the junction area. So when we don't apply any bias, and we illuminate with UV photons from the zinc oxide side, these photons are absorbed by zinc oxide, which is a wide bandgap semiconductor. And then we have photoelectrons flowing from zinc oxide to silicon, and this is the negative photocurrent that we observe here. And when we apply visible photons, these are absorbed both in zinc oxide and in silicon. Silicon obviously absorbs visible photons, but zinc oxide too, despite being a wide bandgap material, from photoluminescence measurements here on the right, we see that due to defect states, it also absorbs a little bit in the visible. So in this case, we have electrons flowing in both directions, both from zinc oxide to silicon and vice versa. And this means that the net current depends on which component is stronger. And actually at 500 nanometers, it starts reversing, which means that silicon photocurrents start to dominate and the photocurrent tends to become positive. And indeed, then when we go to infrared photons, these are exclusively absorbed in silicon because now zinc oxide is a transparent window. And in this case, we have these electrons transported we have transported by the internal electric field, and we have the positive high photocurrent that we observe. Now, when we and this is the final response. So we have very low photocurrents, in the low wavelengths, and then at different wavelengths where we have absorption in silicon, we get a high photocurrent. But when now we apply a negative bias, now we have an external electric field opposing the internal field, which is weak because both materials are n-type. So this is a weak internal electric field. So now, no matter where the electrons are generated, which means no matter what wavelength we illuminate with, they're always transported in the direction of the negative photocurrent by the external electric field. And indeed, in this case, no matter what wavelength we employ, we always have a high responsivity for all wavelengths, which is induced by the external electric field. And this was a very, very interesting observation because with photodetectors, one key component is that to be able to tune them depending on the application. This is a very important request from photodetectors. And I'm going to go to the last part of my presentation now, which is about structuring to make smart surfaces, starting with the surface wettability. Surface wettability is important for various applications in several materials. And to start with the fundamentals, wettability means whether a surface is hydrophobic or hydrophilic. A hydrophobic surface, in a hydrophobic surface, water droplets create high contact angles above 90 degrees, okay, for example, the lotus leaf. And on a hydrophilic surfaces, water spreads, so the contact angle is low, below 90 degrees. So here, water spreads on hydrophilic and remains as a droplet on a hydrophobic surface. And one factor that greatly affects the wettability of a surface is roughness. One surface can be nanostructured or microstructured, but recently it has been found that actually hierarchical structures combining both micro and nanoscale structuring are very interesting for wettability studies. So in our case, we created hierarchical surfaces by growing zinc oxide nanorods on microstructured silicon surfaces. So we have now the silicon microcones, and on top of them, we have the zinc oxide nanorods. And these surfaces, when they are, as grown, they are super hydrophilic, so water entirely spreads on them and the contact angle is very low. But when we anneal them to like 3 different temperatures here, they switch to super hydrophobic, and the higher the annealing temperature, the more hydrophobic they become. And at the same time, they're also highly adhesive. So the droplet does not roll off the surfaces, it stays in place. So here we see such a surface after annealing. It's really hydrophobic because we see that the water droplet remains a droplet, it does not spread on the surface. And then what we're going to do is that we're going to start rotating. Now we rotate the surface up to 90 degrees and the droplet stays in place. It does not go anywhere. So it's both hydrophobic and adhesive. Then now we are even going to start moving it by hand, just by, lifting it up, and still the droplet stays in place. It doesn't want to go anywhere. So these are really adhesive surfaces. But more importantly, we found that when annealing at the highest temperature we tried, which was 400 Celsius, the surface was not just hydrophobic, it was entirely water repellent. So here we have the surface annealed at 400 C, and we're going to start to try to deposit a droplet. And we see that even when we push, the water droplet does not want to adhere to the surface, but it prefers to climb up the pipette that we used. And this behavior can be reversed if we irradiate the surfaces with UV radiation. So we had hydrophilic when we started, and then they switched to hydrophobic when we annealed. And then if we radiate with UV radiation for several hours, at the end they become super hydrophilic again. And this is here the comparison without and with UV radiation. This is important. All these are important for digital microfluidics, where one now starts processing droplet by droplet the fluids on the digital microfluidics. And with these surfaces, we can induce directed liquid motion, frictionless liquid transport, because they are so repellent that you're not going to lose any water at the walls of the microfluidic channel. And going to the last part of my presentation, which is the work that we did also at VULRC in the Lasers for You facility. We also try to do some surface patterning for antibacterial surfaces. So my colleague, Theodoros Giannakis, then a PhD student, now Dr. Theodoros Giannakis, visited the facility at VULRC, and the project was to nanostructure silicon for antibacterial action. So in order to nanostructure silicon, they employed a dye-sapphire laser, a femtosecond dye-sapphire laser at the VULRC, which was focused on the silicon sample, which was in a cuvette filled with water. On a translation stage for processing of a big area. And we see here that after irradiating properly the silicon surface with femtosecond laser pulses in water, we achieved this nanostructuring on the surface of silicon. And now, by combining now this nanostructure with microstructures that we make in our laboratory, we're able to measure antibacterial action on 3 different silicon morphologies: flat silicon, microsilicon with a nanosecond laser here in the middle, and nanosilicon with a femtosecond laser produced at VULRC here on the right-hand side. Here at the bottom is a picture of how the bacterial colonies look like. We studied the E. coli bacteria, and these are the colonies, and then we can measure how many of these bacteria survive or they are killed by the surfaces. And we see that when we use flat silicon, there is no antibacterial action, so all bacteria remain alive. And nanostructured surfaces are pretty good. They kill 61% of the bacterial population, but the nanostructured surfaces are even better, and they were able to eliminate 81% of the bacterial population. So these are our first results, and we're now in the process of studying more and understanding more the mechanisms of this effect, and hopefully soon we will be able to publish this and share it with everyone. And with that, I would like to thank my team members and collaborators who worked on all these topics, students and postdocs, and of course to acknowledge collaborations and funding, collaborations from Greece and abroad on all the topics I mentioned. I would especially like to thank our collaborators at Vilnius, Dr. Ona Balatsnaite and Dr. Virgilius Vaikaitis, and of course the funding that we received from the Lasers for EU facility to visit the premises at VU LRC and perform our experiments. And I would like to thank you for your attention and invite any questions you may have.